Chapter 3, the fourth video lecture here. We're going to continue on with, go away, talking about the different layers of the bacterial cell envelopes. Keep in mind, again, we're talking about bacteria. Now, first of all, if you're working through your Learn Smart Labs activities, your virtual lab activities for this part of the course, you will encounter bacterial staining, where you are virtually applying dyes to bacteria so that you can visualize them better with the microscope. And you'll be learning about how the gram stain is the most commonly performed type of bacterial cell staining. And this was actually developed back in 1884 by Hans Christian Gram. So developed over 130 years ago and still used today. Now back then he didn't really understand how this staining process worked. He just knew that it worked, and it tended to divide bacteria into two major categories. One category that we call gram positive, and another category that we call gram negative. So it's a multi-step process to perform a gram stain in the laboratory. It's not too hard, but you've got 4 different chemical reagents that are used. 2 of those are dyes. One's called crystal violet, which has a dark purple color, and the other one is saffronin, which has a reddish pink color. And based on the cell envelope or cell wall properties of bacteria, at the end of that process, they either come out purple or they come out a reddish pink color. If they're purple, they are gram positive. If they are pink, they are gram negative. So you want to remember that. All right, so we just said that the cell envelope or cell wall of gram positive and gram negative bacteria determines whether or not they come out as gram positive or gram negative. And you're going to hear this terminology used when you're around a clinical microbiology laboratory. Whether or not a particular bacterial organism is gram positive or gram negative is one of the key things that we use to characterize them. And to begin the process of identification, it helps us narrow down identities. Okay, so what are the characteristics of a gram positive cell wall? Okay, so first of all, now we talked about in the last video lecture peptidoglycan, the peptidoglycan cell wall. So in a gram positive cell wall, that peptidoglycan layer is very thick, and it's colored purple here on this diagram. All right, and again, keep in mind this diagram makes it look like that's a thick brick wall or something around the surface. No, it's a rigid structure, but it's very porous. But nevertheless, it's going to protect that bacterial cell from bursting if it's taken up water. And then inward from there, so this would be the inside of the cell over here, of course. Inward from there, that's just your cell membrane or cytoplasmic membrane, plasma membrane. All of those terms are referring to the same thing. So you've got this thick peptidoglycan layer that's outward from the cell membrane. All right, now there are a couple of other compounds that you find in gram-positive cell walls, but not in gram-negative. Those include teichoic acids and lipoteichoic acids. So some teichoic acids are called wall teichoic acids, so they're just embedded within the peptidoglycan. And lipoteichoic acids have a lipid portion down here at the bottom. And that lipid portion, remember lipids love other lipids. Hydrophobic molecules love other hydrophobic ones. And so the lipid portion of a lipoteichoic acid loves to embed itself down in the phospholipids of a cell membrane and helps anchor it there. So they're thought to help, those lipoteichoic acids are thought to help anchor the cell wall to the underlying cell membrane. Sometimes those teichoic acids and lipoteichoic acids can help bacteria stick to tissues somewhere, kind of like the fimbriae. When we were talking about fimbriae back on the first video lecture for chapter 3, I guess that was the second one. Those are generally found on gram-negative bacteria, not gram-positive. So teichoic acids and lipoteichoic acids are something that gram-positive bacteria can use to help them adhere to tissues. Your immune system sees these as foreign molecules. So that's a concept we want to keep thinking about in microbiology is, some of these molecules are bacterial molecules. Our bodies don't make them, and your immune system, which is the key thing that fights off microorganisms that could potentially harm you, is it's looking for things that are foreign to your body. And so teichoic acids and lipoteichoic acids are examples of those. All right, and notice down here on the at the bottom of the slide, relate to gram stain outcome. What do I mean by that? What I mean by that, when you guys are doing your virtual gram staining activity is for homework, you get cells that are purple at the end or pink. So purple ones are gram positive. So when you see gram positive cells, you need to think, okay, that means they've got a thick peptidoglycan layer. They contain teichoic acids and lipoteichoic acids. Those are kind of the key things that we would associate with a gram-positive cell wall. Now, how about a gram-negative cell envelope? What's going on there, or cell wall? So this is a diagram of a chunk of a gram-negative cell envelope. One key thing, if an organism is gram-negative, it has a thin peptidoglycan layer. And also think about the color. If you were doing a gram stain, and you guys will be doing that in your virtual labs, the cells come out pink or reddish pink at the end. Okay, and so if that's the case, then that organism has these particular characteristics of its cell envelope. So a thin peptidoglycan layer, not nearly as thick as what we saw with gram-positive. Then another major difference is gram-negatives have an outer membrane. A second lipid bilayer membrane. So what is a lipid bilayer? A lipid bilayer means you have two sheets of lipid molecules, kind of a lipid sandwich that has formed to create a membrane. And so if you guys have had any prior biology class, you've learned before about lipid bilayer, two-layer membranes composed of phospholipid molecules. Okay, so an outer membrane has two sheets, and the inner one, right in here, I'm kind of drawing across, that's built from phospholipids. And that's like what you learn about when you take A&P or freshman biology, the phospholipid membrane bilayer. This outer layer, though, contains phospholipids, but also special molecules that are called lipopolysaccharides, LPS. So that name tells us that they are combinations of lipids and polysaccharides. Polysaccharides are chains of sugars. And the lipid portion is actually called lipid A. That's a special gram-negative bacterial cell lipid that it's not produced in the human body. It's a special gram-negative bacterial cell lipid called lipid A. So you have these LPSs, lipopolysaccharides, in the outer leaflet, that outer layer of the outer membrane of gram-negative bacteria. I'm going to erase all of that. I want to draw your attention to lipid A again. This is sometimes called endotoxin. And we'll talk more about that later in the semester, but it's mentioned in your textbook here. And lipid A, again, that is a foreign molecule to the human body. Bacteria make that, we don't. And gram-negative bacteria make it, we don't. When your immune system detects lipid A, it sets off alarm bells. It lets your immune system recognize, oh, hey, there's some gram-negative bacteria here, and we need to bring in the forces and fight them. However, sometimes your immune system overreacts to lipid A, and it can actually be toxic for the human body. It can actually be so toxic it can wind up killing a patient if they have these gram-negative bacteria spread all throughout the body. Like, let's say you have a blood infection with gram-negative bacteria. So because of that, lipid A is sometimes also called endotoxin, because it's a toxin that is within the structure of the bacterial cell, and it's actually when the bacterial cell dies and breaks apart that those lipids are released and set off this cascade of reactions where your immune system is overreacting, and in some cases that can be toxic and even deadly. And we'll talk more about endotoxin when we get further into the class later in the semester, but I wanted to go ahead and point that out now. And also read about it in your textbook as well. All right, another feature of a gram-negative cell envelope, these things that look like barrels on the diagram, those are called porins, and those are big protein channels that allow substances to pass from out here toward the inside of the cell. They create channels that allow things to pass through. All right, so one thing I want you guys to think about is that gram-negative bacteria have these two membranes. So they've got this outer membrane that is an extra barrier at the surface of the cell. And the porins are more or less governing what's going to be allowed to pass from the outside to the inside. So gram-negative bacteria tend to have this little extra membrane layer of connection, protection that keeps substances out, and those substances can include things like antibiotic drugs, for example, or disinfectants or antiseptics that you're using to try to kill bacteria. In a lot of cases, gram-negative bacteria are more resistant to those types of things, and so that's going to come up over and over again as we move through the semester. Another term that you may see related to gram-negative bacteria, periplasmic space. So between the plasma membrane, cell membrane, and the cell wall, and over here between the outer membrane and the cell wall, you've got a jelly-like space that's called the periplasmic space. And that space includes quite a few digestive enzymes. So if you've got large molecules or fairly large molecules that were able to pass through the porins, those are pretty big channels. They wind up in that periplasmic space, and then the gram-negative bacteria have digestive enzymes there that will chop them up into smaller molecules that can then pass across the cell membrane and go inside the cell where they can be used as nutrients. All right, so some key things about gram-negative cell envelopes that we want to remember. We want to remember that they have a thin peptidoglycan layer, whereas gram-positive have a thick peptidoglycan cell wall. And they have this outer membrane. Gram-negatives have that, gram-positives do not. And the outer membrane contains a special lipid polysaccharide combination molecule called LPS, so that's something that you only see with gram-negative bacteria and not gram-positive. The lipid portion of that molecule is called lipid A, and lipid A is sometimes referred to as endotoxin. So you'll hear that term again used quite a bit. Gram-negative bacteria have porins in their outer membrane. Those are these barrel-like protein channels that allow some substances to pass across. And then finally, they've got that periplasmic space, kind of a jelly-like filling on either side of the cell wall where you've got some digestive enzymes present. So your gram-negatives don't have the teichoic acids, they don't have the lipoteichoic acids. Those were gram-positive cell wall features, nor do they have the thick peptidoglycan layer. So you kind of want to make a chart of gram-negative versus gram-positive cell envelope, cell surface, and what features each of those have. And study your diagrams. On the test, there will be pictures of these gram-negative and gram-positive cell surfaces, and so you want to be able to recognize those different features that are there. All right, here's a diagram again that's comparing the gram-positive versus the gram-negative cell surfaces. And so I think I just kind of verbally went through everything. But again, that's a good thing to study and make your comparison of. Okay, so there are some exceptions. So most bacteria out there on Earth that we know about either have a gram-positive cell envelope or a gram-negative one. However, there are some exceptions, and some of them are pretty important. All right, one exception are the Mycobacteria. Okay, so these are bacteria that have cell walls that are not built of peptidoglycan. Instead, they're built from a compound called mycolic acid, which is actually a waxy lipid. So it's almost like they've got a very thin veil or covering of wax at the surface. And unfortunately, that waxy mycolic acid that they have in place of peptidoglycan. Now, we already said peptidoglycan is pretty porous. It's not much of a barrier, but mycolic acid is a pretty tight barrier. So that mycolic acid actually protects those bacteria from things like drugs and antiseptics and disinfectants being able to penetrate and go inside and kill the cells. So that's an advantage to the bacteria that have them. So we're going to hear about that quite a bit during the semester. The mycobacteria come up a lot. And the most important example, medically important example, Mycobacterium tuberculosis. So Mycobacterium is a genus name and tuberculosis is the species. Obviously that's the bacterial organism that causes TB or tuberculosis. And one of the, probably the most successful human pathogen on the planet. I think I recently read not too long ago that tuberculosis has killed more human beings than any other human pathogen and continues to be a major health problem on the planet. Not as much of a problem in the United States. We're very good at controlling tuberculosis. However, it is still a problem, especially when you get into homeless populations and other very poor populations where people don't have good access to health care, live in conditions of poor sanitation. AIDS patients who have very weak immune systems are more likely to get tuberculosis. And then we also have drug-resistant strains of tuberculosis that have emerged, which are threatening to bring tuberculosis back and make it more common out in the population. I do want to make you guys aware of something called the acid-fast stain, and that is a special type of stain, kind of like the gram stain where you use two dyes, a purple one and a pink one, and bacteria come out either purple or pink at the end, and they're called gram-negative or gram-positive. An acid-fast stain is another staining process with two dyes, and the mycobacteria come out pink. Other bacteria end up blue at the end of the staining process. And so let's say you take a sputum sample from a patient who is suspected of possibly having tuberculosis. You can do a quick stain in the laboratory. You can do an acid-fast stain. And if you look in there, and kind of like this picture is showing you, you see all these pink bacteria in that sputum sample from the patient. That's a very good sign that they are infected with Mycobacterium tuberculosis. Another relative of Mycobacterium tuberculosis is Mycobacterium leprae, which causes the disease leprosy, another ancient disease that has afflicted mankind for a very long period of time. You're unlikely to see lepers, though, in your medical careers. Not a significant health problem anymore in the United States. There's another group of bacteria called the Mycoplasmas. These are teeny tiny bacteria, really, really small. They don't even have a cell wall. So they're kind of like us, no cell wall. They have a cell membrane. Their cell membranes are reinforced, made a little bit more robust with special lipids that are called sterols. And sterol may remind you of the word cholesterol. And you may have learned in anatomy and physiology or a freshman biology class that cholesterol is a special type of lipid that exists in the cell membranes of animals, animal cells like ours, and it helps make the cell membrane more robust, protects it from falling apart too easily. So mycoplasmas don't have a peptidoglycan cell wall, so instead they have these sterol, special sterol lipids in their cell membranes that reinforce the cell membrane. That helps protect those cells from bursting due to osmosis when cells take up water. An important pathogenic member of that group, Mycoplasma pneumoniae or pneumoniae. You'll look carefully at how that word is spelled. Pneumonia just has an A on the end. If you stick an E on there on the end, it changes the pronunciation. So now it's pneumoniae or pneumoniae. Those are the two Latin pronunciations for that word. And that organism, if you or you know somebody who ever had walking pneumonia, that's a milder type of pneumonia, but it takes a long time to get rid of. Mycoplasma pneumoniae is typically the type of bacteria that causes that illness. All right, so that concludes this particular video clip. The fifth one for Chapter 3, we're going to finally get to the cell membrane and talk a little bit more about what's going on at the cell membrane that covers the surface of bacteria.